Alright, so we're going to begin chapter 4 now. Now you'll notice we have skipped chapters 2 and chapters 3 in the textbook. That's not to say that they're not important, but they go over things that we just felt like you should already know or that you will learn in lab. So if you want to go back and look at those chapters, especially if it's been a while since you've taken biology, then you may want to do that. Chapter 2 simply goes over cells and the chemistry of those cells. Chapter 3 looks at lab, which we're going to hit on when we actually get to the lab component of the class. So if we skip to chapter 4, we start breaking down that list. Now remember, there's a list in chapter 1 that you have to know, and so these were microbes or microorganisms. And so the first one on that list was bacteria. Now that was not a coincidence that it was the first one on the list. It's the first one on the list because we will spend a majority of our time this semester dealing with bacteria. Now if you remember, bacteria fall under the category of prokaryotes. So in chapter 4 we're going to look at prokaryotic cells and the microorganisms that fall in this category. Now let's remind ourselves, as far as cells are concerned, whether they're prokaryotes or they're eukaryotes, all cells share some common characteristics. Talk about this in biology. Cells have basic shape. They have basic internal content, so they're going to have cytoplasm, right? And then all cells are going to have genetic material, so DNA. They're all going to have ribosomes. Even if they're a prokaryote, they're still going to have some much smaller and simpler, but they're going to have ribosomes. Now remember, cells are either eukaryotes or prokaryotes, all of this kind of a review of stuff that we've already looked at in chapter 1 or in biology. And then also in biology, we talk about some basic characteristics of life, so of cells. We know that cells are going to reproduce. That's the only way that you get new cells, right? Remember mitosis that we talked about in biology. Even some cells do meiosis, right, for sexual reproduction. We talk about the fact that cells are going to grow and develop. Cells are going to have chemical reactions, so they're going to have metabolic reactions. Now, whether they're breaking things down or building things up. We know that living things are going to move. They're going to be stimulated to move or to respond, either with internal environment or external environment changes. We know that living things are going to have support, so they're going to have structures in place to support them, to protect them, to store. And of course, living things are going to have some form of transporting nutrients in and around, and then waste materials out. So again, basic structure of a prokaryote is what we're going to look at in this chapter. So remember, this is made of a cell. Prokaryotes will typically have a cell wall. They'll typically have a cell membrane. And then of course they'll have genetic material that does not have a nucleus around it. You'll see ribosomes scattered. And then some storage vessels that we call inclusion bodies. Also a lot of bacterial cells will have either an appendage or multiple appendages on the outside. So if we look at these things a little bit more in detail, we're going to work our way external to internal. So we're going to start looking at these structures specifically from the outside towards the inside. So if we look at the outside, the first thing that we'll come across on a microbe, specifically a prokaryotic microbe, are some external appendages. Now, if we get specific here, external appendages are typically going to be used for either motility or attachment. So if we're kind of distinguishing these appendages, when we look at motility, obviously that's the word for movement. A lot of microbes, prokaryotic microbes, will have flagella for movement, or they'll also have some extra filaments that kind of help them to move. As far as attachment is concerned, a lot of microbes will have fimbriae, which they use for attachment. Pili they can use for attachment as well, and the pili itself, when it attaches, it can also be used as a channel to transport materials. We also mentioned glycocalyx here, can be used for attachment as well. So again, if we get a little more detail, let's look at flagella first. Now we know flagella, it's one of those appendages that's used for motility. And so if you look at a basic flagella, it's going to have a filament, which is this long, thin structure. It's going to have some type of hook or curved area that's covered with a sheath, and then it's going to have the basal body that's here. Now, the main thing to note about flagella, again, is it's going to function in motility. So it's going to function in movement. And as far as flagella are concerned, we know that some eukaryotic cells have flagella, But when a prokaryotic cell has a flagella, the movement of it is different than the way that it's found in a eukaryote. So for prokaryotic flagella, it's going to rotate 360 degrees. So it's going to move like a propeller as it pushes that cell through its environment. So it's an important thing to note. They mention here that some microbes will have one flagellum, some will have multiple flagellums, some have on one end, on both ends. These are terms that I would not be too concerned with knowing, but just note that they can have different types of flagellar arrangements. And then the last thing I want to mention about flagella here is an interesting concept that most prokaryotes have in terms of using their flagella. So in a lot of cases, their flagella will respond to either chemical stimuli or to light stimuli. And so two vocabulary words here they mention, chemotaxis and phototaxis. So chemotaxis is flagellar ability to respond to chemicals. And so as they sense a chemical, if they think it's dangerous, then they'll use negative chemotaxis to move away from that chemical, or positive chemotaxis to move towards that chemical, maybe if it's food. And then they also use phototaxis, which is just the ability to respond to or towards light. So flagella, one of the appendages that prokaryotes can have. They also can have pili, which is the plural form, or you can use the singular form, which is called pilus. So if we look at pilus, it's basically just a tubular structure. It's hollow because it's a channel that's used. Now, typically it's only found in gram-negative bacteria. And what it does, you'll notice in the picture, is it will join one bacterial cell to another. And so this long pilus or tube will connect, and then what the microbes are able to do is to exchange genetic material. So this pilus is actually useful in a form of reproduction that we call conjugation, where they literally exchange genetic material and they use that to reproduce. A third appendage that they can have is called fimbriae, or fimbriae. And so these are hair-like bristles. A lot of people confuse these with cilia, which a lot of eukaryotic cells will have. But fimbriae, they're more hair-like bristles, if you will. They're going to extend from the surface, and their main function is going to be in adhesion. So it helps them to stick to other cells. It will also help them to stick to a surface. So in our case, if bacteria gets in our system and it has fimbriae, it can use those fimbriae to adhere to the lining of certain cavities so that we don't flush it out of our system. And then, of course, it also mentions the glycocalyx, which not necessarily is an appendage, but it is an outer layer, and so we kind of mention it with the appendages. And so the glycocalyx by structure is made of sugars and proteins, so we typically call it just a sugar coating. It can be found in a very loose organization, which is called just a slime layer, or a very highly organized, tightly compact layer that we would call a capsule. But either layer, it's referred to as the glycocalyx, and it's sugars and proteins. Now, purpose of this glycocalyx, there's a couple of reasons why a prokaryotic cell would want to have them. One is for protection, because just having that sugar coating on the outside, it keeps the cell from getting dehydrated, also keeps them from losing important nutrients that they need to hold on to. But a big benefit to the glycocalyx is cells that have them, it can inhibit white blood cells from actually eating them or phagocytizing them. We know that for human bodies, our first response to a foreign object being in our body is we try to phagocytize it. We try to send white blood cells to that area and eat it. But if they have that sugar coating, in a lot of cases, it will keep them from getting eaten, which in turn will allow them to continue to reproduce and potentially cause infection. And then, of course, the sugar coating can also be used for attachment. You think about the fact that sugars can be sticky, then they can use this sugar layer to adhere to other cells. And so what actually happens in a lot of cases is when they have this glycocalyx, they'll form layers of microbes, which we call biofilms. And so a lot of times you can see these biofilms, they'll form on things like a catheter. They also can form, like dental plaque is an example of biofilms, where you get lots of layers of microbes all adhering in one specific area. And so that's where we'll stop part A of